It's our last topic for Bio 111, and today we're going to talk about the senses. We'll start with the general senses, and then we'll look at the special senses. We're going to leave vision, however, until next term, because that's a whole topic just on its own. Of course, we can't survive without these senses. We need them to identify dangers, but also to identify opportunities. Sensation is the biological awareness of changes in the internal or external environment. Now, note the word biological. We could be talking about changes in blood pressure or changes in blood pH. That's not something that you are consciously aware of. There's a biological awareness in that your body, your brain, is aware of what's going on, but you yourself, as the person you are not necessarily aware of this change. Perception, however, is the conscious interpretation of these stimuli. So you're being stimulated, you're learning something about your environment, and that is being consciously processed. Special senses are the result of specialized sensory structures that are found in special sensory organs. So for instance, within the nose, within the olfactory epithelium of the nose, you have specialized cells that can detect the shape of molecules. And that's interpreted as smell, but the structures are specialized just for this one sense. The eye is highly specialized. As I mentioned, we're not going to talk about that this term, but the retina contains cells, rods and cones, that absorb photons of light. And then that information is sent back to the brain. But that's all the eye does. It just collects visual information. The tongue, highly specialized for taste. So we have taste buds that contain receptor cells that are sensitive to very specific modalities of taste. And we'll talk about what that means a bit later. Within the ear, we have highly specialized structures that can sense pressure changes within the air, basically, sound waves. Now, we also have general senses. General senses don't require a highly specialized organ. So we have these sensory neurons that can be distributed throughout a membrane or throughout a tissue layer. The best example would be the skin. So within the skin, we have a collection of different sensory neurons that are distributed all over the place. We have pain receptors, we have touch receptors, we have pressure receptors, we have temperature receptors. Again, the take-home message here when we're talking about general senses is that those receptors aren't all clustered together in a specialized organ that only does one thing. Also, we have proprioception going on. We talked about that before. That's the sensation or perception of where your limbs are in three-dimensional space. So that's accomplished by things like stretch receptors in muscles that tell your brain whether a muscle is stretched or contracted. And that will help your brain figure out the position of your limbs. We have chemoreceptors that will sense the pH and chemistry of the blood, for instance. That would also be a general sense. Receptors, the specialized cells that respond to the environment and send off an appropriate signal, can be classified based on where they are or what they do. Let's start with where they are. We have externoreceptors, which respond to the external environment. And these are found in the skin, of course, but they also make up some of the special sense organs. The olfactory receptors in the nose are responding to air that you're breathing in from the outside. These would consist of touch, pressure, pain, temperature, and as I mentioned, some of the special sense receptors. Then we have ontoreceptors. These respond to the internal environment. These are receptors that are found in the viscera, the internal organs, and also within the blood vessels. So they respond to pH, carbon dioxide, oxygen, other chemical inputs from the blood and interstitial fluid, temperature, pain, pressure, and stretch. So of course some of these you're consciously aware of, some of them you're not. Then we have proprioceptors, which are going to respond to the body's position and motion. So stretch receptors are found in skeletal muscle, tendons, ligaments, and other connective tissues, and they'll tell you if a certain tissue is under tension. And your brain can interpret that and figure out what your limbs are doing. We also have the vestibular system within the ear, which is responsible for equilibrium. It tells you if you're standing on your head, if you're standing upright. It tells you about the position of your head relative to the rest of your body. Now let's classify receptors based on what they respond to. First of all, we have mechanoreceptors. They're going to detect mechanical deviation or distortion. If there is some sort of change in shape, if something is being stretched or compressed, if there's some sort of physical force being applied, then a mechanoreceptor might be able to detect that. They detect touch, pressure, vibration, and proprioception. Then we have thermoreceptors. Thermo meaning temperature. These are receptors that sense changes in temperature. We have nociceptors, which is a fancy name for a pain receptor. They are going to respond to physical or chemical damage. Photoreceptors are found within the eye. These are the rods and cones of the retina, and they're going to respond to photons of light. And then we have chemoreceptors that respond to molecules and chemicals. They detect the shape of different molecules, and they're responsible for taste, smell, and also for detecting the pH and chemistry of the blood. The skin is how you make contact with your outside world and how you interact with your world. So it's not terribly surprising that there are a lot of general sensory structures within the skin. We have free nerve endings, and these are naked, uncovered nerve cells. The dendrites of these extend up into the epidermis, so they come quite close to the surface. They can be damaged, and that can result in the sensation and perception of pain in your brain. But they can also respond to tactile forces, so touch, to temperature as well. You have root hair plexuses, and this is where you have nerve cells that wrap around the base of a hair. If someone touches you very lightly and they just move one of the hairs on your arm, let's say, what's going to happen is the movement of that hair is going to stretch these cells and cause them to depolarize and send off an action potential. So they're very sensitive. We also have what are called Merkel cells. Merkel cells are found within the epidermis, predominantly within the stratum basal. And they're sensitive to very, very light touch. Deeper down into the dermis, we have tactile corpuscles, which respond to pressure. And deeper down still, we have larger structures known as lamellated corpuscles, also known as Pacinian corpuscles. They have several layers of collagen wrapped around them. So these cells, in order to be activated, in order to be triggered to give off an action potential, they need to be distorted by a fairly powerful force. So these are going to respond to very deep touch or to deep vibration. We also have Ruffini corpuscles, and Ruffini corpuscles are sensitive to distortion and stretch. If someone pokes your skin, that is going to cause your skin to depress, but also to stretch out. Think of your skin like the surface of a trampoline. If you stand in the middle of the trampoline, you stretch that membrane. And these are sensory structures that are going to be activated by that stretch. These receptors that we just talked about can be grouped into two broad categories. We have unencapsulated and encapsulated. And it's exactly what it sounds like. So if they're unencapsulated, then they're not surrounded and protected by layers of connective tissue. If they're encapsulated, then they are, and it takes more force to cause them to distort sufficiently to fire off an action potential. So free nerve endings would be a prime example of an unencapsulated receptor. Remember, those free nerve endings can extend up through the cells of the epidermis. We have these Merkel cells that sit on top of tactile discs that are also unencapsulated, generally found within the stratum germinativum. Hair follicle receptors or root hair plexuses, whichever term you want to use, are also unencapsulated because we have these free nerve endings that wrap around the base of the hair. Encapsulated receptors are encapsulated by layers of connective fibers, collagen mainly. And that means it takes more force to distort these cells and trigger off an action potential. The Pacinian corpuscles are probably the best example of this. They have many, many layers of collagen. But we have Mesner's corpuscles, also known as tactile corpuscles, Pacinian corpuscles or lamellated corpuscles, Ruffini's corpuscles, and muscle spindles. These are all excellent examples of encapsulated receptors. If you think back to when we first started talking about nerve cells and how they worked, I mentioned the different ways that a cell might be brought up to threshold to bring about an action potential. That original depolarization is generally caused by the influx of sodium. And remember that mechanoreceptors consist of a channel that basically has a little lid on it. And if the membrane that channel is embedded within is stretched, that little lid opens up. It allows sodium in. That brings the cell up to threshold and it fires off an action potential. That's what happens in most of the mechanoreceptors we've been talking about. Now what you're seeing here are stretch receptors, which are found in muscle. And if the cell is stretched, so if those muscle cells are stretched, this mechanoreceptor is also going to be stretched. Basically, we have the ends of the nerve fiber wrapped around this tube, and when that tube stretches out, it stretches out that nerve cell, and it causes those little gated channels, those little channels with a little hat on the top, basically, to open up. So the little cap on the top is going to pop open, it's going to allow sodium in, and the cell is going to fire off an action potential. And that will tell your brain that that particular muscle is stretched. Nociceptors are pain receptors, and they can be activated by a number of different stimuli. They can react to direct damage, or they can react to damage in the cells around them. The diagram on the left here gets into a lot more detail than we're going to cover, but you can see there are many different receptors. That's shown on the right of the table, and there are many different stimuli that can bring about a triggering of one of these cells. So serotonin and also the first two compounds on our list there are things that might be floating around in the interstitial fluid if there's been damage to nearby cells. Those are things that would not normally be in the interstitial fluid. The same with ATP. ATP is used as the currency, the energy currency of a cell. It doesn't leave the cell unless the cell is broken. So if we have a receptor that's triggering the presence of ATP in the interstitial fluid surrounding the cell, then that probably means that a lot of cells have been broken. And that is going to be interpreted as pain, as damage to your body. Likewise, if there's lots of lipids floating around, probably some cells have been busted open. If there's lots of hydrogen protons, then we've got an acidic condition. That's a damaging condition. And of course, if there's high pressure, that means we have increased blood flow to the area. That's a sign of inflammation. If we have lots of heat, that's also a sign that damage is probably being done or could be done. And I mentioned the TRPV pain receptor before when we talked about neurons and how they work. This is another example of a receptor that can react to many different things. These pain receptors are reacting to signs of damage or reacting to changes in the environment that could bring about damage. Baroreceptors detect changes in blood pressure. And we have these within the aortic arch and also within the carotid sinuses. They're going to send signals up through the glossopharyngeal nerve to the brainstem. And then from the brainstem, we have effector signals that come out through the vagus nerve, and they can adjust the heart rate appropriately. In addition to the baroreceptors that are found within the carotid artery and the aorta, we have these structures known as carotid bodies and aortic bodies. These are clusters of chemosensory cells, and they're going to send information back to your brainstem. They're constantly monitoring the chemistry of your blood, most notably the amount of dissolved oxygen, the amount of dissolved carbon dioxide, and also the concentration of hydrogen protons, which of course indicates pH. Your brain is going to take appropriate responses without, of course, you being consciously aware of this. You may be aware of the outcome though. So for instance, if your carbon dioxide levels are really high in your blood, you may start panting. And what panting does is it gets rid of that carbon dioxide. Typically, if you have a whole lot of carbon dioxide in your blood, it's an indication that the blood has become acidified. We'll talk about that more next term, but if you have a lot of hydrogen protons in your blood, there's a buffering reagent that takes those hydrogen protons, adds them to oxygen to make water, and in the process also makes carbon dioxide. But like I said, we'll come back to that next year. These receptors within the brainstem also monitor your breathing rate. So we have stretch receptors within the lungs that carry information back to the brainstem. Those are going to indicate how quickly you're inflating your lungs and how much you're inflating your lungs. We have irritant receptors in the lungs and also within the carina, which is where the trachea branches into the two primary bronchi. And that will send information back to the brainstem that can bring about coughing or gag reactions. Over the summer, actually, I was hiking and I sucked in a mosquito. I breathed it in and it actually hit the carina, and it's actually kind of painful. This is coughing, very deep coughing that you can't do anything about. You have no choice in the matter. Sensory receptors become less responsive to a stimulus if they're constantly stimulated. And this is a process known as adaptation. Now, this occurs with all of the sensory receptors with the exception of pain and proprioceptors. Probably the best known example would be smell. I know Febreze used to have an advertising campaign where they would say, "Have you gone nose blind?" And the idea was that if you're in a room that smells bad, for only a few minutes, you don't notice it anymore. Someone else walks in that room, they notice it. The sense of smell adapts quite quickly. Now, pain receptors keep sending off a signal. If they're sensing damage or they're sensing a harmful environment, they will constantly send a signal back to the brain. Now, if you are dealing with pain over a prolonged period of time, you can become a bit numb to it. But that's not going on in the pain receptors, that's going on in the thalamus of your brain. Your brain is kind of overriding that input. Proprioceptors don't exhibit adaptation either. So no matter how long you've had your arms in a certain position, your body still knows where they are. Your body still knows what position your body is in as well. Chemoreception can be either a general sense or a specialized sense. If we're talking about the pH of the blood and the chemistry of the blood, that would be detected by your general senses. But if we're talking about taste or smell, these are sensations that still require chemoreceptors, but of course they also require very specialized organs. With a chemoreceptor, what's happening is these receptors are responding to the shape of molecules that are dissolved within an aqueous, water-based solution. Olfaction is just a fancy word for smell. And we receive all of the inputs related to smell from a rather small part of our nasal cavities. So right at the top of the nasal cavities, we have this area of about 5 centimeters squared that contains the receptors that are responsible for olfaction. We have 10 to 20 million of these cells. Each one is a specialized neuron that is going to send off an action potential if it's exposed to a molecule that has a certain shape. The olfactory epithelium contains a few different cell types. The most specialized, of course, would be the olfactory cells, which are receptor cells. They're specialized neurons. Now, at the top, on the surface, they have these projections that are known as cilia. Now just note they're not really cilia in that they're not going to beat back and forth and cause movement. Instead, they're stuck within a layer of mucus and they're covered with receptors. So they're covered with receptor proteins that will respond to certain shaped molecules. Surrounding these cells, we have support cells, which do exactly what you might think. They help support the structure and help nourish those neuronal cells. At the base, we have a basement membrane because this is an epithelial layer. And attached to that, we have basal cells. Basal cells are stem cells. They act a lot like the cells that are found within the stratum germinativum of the skin. And what's quite interesting is these stem cells can actually give rise to new olfactory cells. In fact, there's been a lot of studies done in rats and mice, and they found that they completely replace all of the cells within their olfactory epithelium every 2 months or so. That's pretty amazing because usually neuronal cells can't be regenerated. So there's a lot of people that are kind of interested in this because this is one area of your body that's capable of regenerating new neurons. Also, we have what are called Bowman's glands or olfactory glands. And here you can see we have a duct that leads up to the surface. And at the base, we have a bunch of exocrine cells that are going to produce mucus. And that's going to coat this epithelial layer in this wet mucus. These chemoreceptors only work if they're wet. Molecules that are suspended within the air that we can detect as smell are known as odorants. So odorants will make their way through the nasal passages with the air that we breathe in, and if they bump into an olfactory cilium attached to one of these olfactory sensory neurons, then that will trigger an action potential that will travel along axons up to the olfactory bulb. Remember that the top of the ethmoid bone contains these little tiny pores. That's an area known as the cribriform plate, and those little pores are for the passage of these bundles of axons. I should point out that our sense of smell is really kind of pathetic compared to a lot of other animals, compared to a dog or a cat, for instance. They have a much larger olfactory epithelium. Ours is pretty tiny. Odorants can be fairly large molecules, and they can have several bits that stick out with different shapes. And we talk about these sticky outy bits as domains. There are at least 350 different receptors within your olfactory epithelium, and each one of them is going to detect a different shape. And of course, you'll have millions of each type. So take a look at the top example here. We've got a fairly simple molecule. It's got two domains. We've got this one part that's kind of square in shape, and then another part that's rounded. Well, that will set off the receptors that can bind to that square-shaped part. That's receptor number 1. And also it will set off other receptors, other cells that can bind to that rounded part. That's example number 3. And you can go down and you can see that these different molecules have different bits and pieces to them, and they can be kind of complicated. They can have many, many domains, and that will set off a different set of receptors. Now, if we were to show all of this, of course, instead of having 4 receptors shown there, we would have 350 to 400 different shapes. Your brain interprets what the molecule is by the sets of receptors that have been triggered. So in that first example there, your brain is going to look through your memory for a molecule that has those 2 bits to it. Now, of course, your brain doesn't know the shape of the molecule, but it's going to look for a memory that relates to a time when those 2 receptors were triggered simultaneously. And that's why your smell is linked so intimately to your emotions and to your memories. It could be that this is something dangerous and you need to react quickly, or it could be that this is something you want to investigate. Within the olfactory bulb, we have what are known as mitral cells. These cells have long axons that are bundled together and travel back to the brain. That bundle of axons is the olfactory nerve or olfactory tract. Now, these mitral cells have dendrites that come off, and they're going to synapse with the axon terminals of the olfactory receptors. This synapsing occurs within structures known as glomeruli. The glomeruli are receiving information about the same shapes. So you can see down the bottom here, we have different olfactory receptors. Again, there would be at least 350 different types of receptors. They're just showing 5 here. But they've represented them as different colors, so the different colored receptors would be triggered by different shapes. And notice that all of the signals are being sorted to the appropriate glomeruli. So let's say that we have, our red receptor, whatever that senses, it's being triggered. And then we have our green receptors, those are being triggered as well. They're going to send information to the appropriate glomeruli, and then those are going to trigger the connected mitral cells, and they'll send that information back to the brain. And this results in what's sometimes referred to as a combinatorial code. Your brain is deciphering this combination of inputs. In summary, odorants are molecules that break off of food or break off of a flower, whatever. They drift through the air and they find their way to your olfactory epithelium. They bind to receptors that are found on the cilia of the olfactory cells, and you have at least 350 different cell types. Each one of these cell types has a different odorant receptor that will respond to a certain shape. Now keep in mind that for each of these cell types you have millions of cells. That information is going to be transmitted up through the cribriform plate to the olfactory bulb. It's going to be transmitted to the appropriate glomerulus. And there's going to be lots of these glomeruli. That is going to send a signal to the appropriate mitral cell. And then that information makes up this combinatorial code that is transmitted to the brain. But how do these olfactory cells depolarize? Well, once again, it involves a G protein. So if the odorant molecule binds to an odorant receptor protein, it has to have the right shape, of course, then what's going to happen is a G protein is going to be activated within the cell. That activates an enzyme that grabs onto any ATP that's floating around and converts it into cyclic AMP. It cuts off a couple of phosphates and it changes the shape of the molecule. Cyclic AMP serves as a signaling molecule, and it's going to bind to calcium-sodium channels, which will allow calcium and sodium which are in high concentrations outside the cell, to flow in rapidly. The calcium also interacts with chloride channels, and the chloride flows out. So we have an influx of a lot of positive charge and an outflow of negative charge. So this is going to very rapidly depolarize the cell, and that's going to set off an action potential that can be carried up to the olfactory bulb. We've talked about G proteins before. We talked about them when we talked about how hormones work. They're very commonly involved in anything that's receptor mediated. So the activities of hormones and neurotransmitters, etc. So note here in this example, it's a hormone that's binding to a receptor, and that's going to allow the receptor to bind to this G protein. The G protein is going to utilize GTP, guanosine triphosphate, as an energy source, much the same way that many molecules use ATP as an energy source, and that will allow this G protein to bind to and activate an enzyme that converts ATP into cyclic AMP. And then that cyclic AMP floats around inside the cytosol of the cell and brings about a lot of changes. Now, I don't expect you to know a lot of detail from this. Just realize that the binding of the ligand is going to allow the receptor to activate a G protein. The G protein is going to activate an enzyme that manufactures cyclic AMP. That's the level of detail I would expect you to know. And if you're wondering what makes cyclic AMP different from ordinary AMP, and I'm sure you are, well, this diagram shows what's going on. So we have an enzyme that's going to be bound within the inner leaflet of the plasma membrane. That enzyme is activated by a G protein. And then what that enzyme is going to do is it's going to cut off two of the phosphates. So it'll break that bond there. And then the other thing it does is it will form a bond between this oxygen and this carbon on the sugar that makes up the resulting AMP. And the fact that we have two bonds introduces basically a loop into the molecule, and that makes it a cyclic molecule. Humans can recognize a little more than 10,000 different odors. However, we're rather poor at determining the intensity or concentration of an odorant. Other animals that can smell a lot better than us, they have a larger olfactory epithelium. That makes it easier for them to determine differences in concentration, and also they tend to have a greater diversity of receptors. They are sensitive to a greater number of shapes on the odorant. Olfaction undergoes adaptation really quickly, more so than the other senses. Also keep in mind that the subtleties of taste come from smell, and you probably know that. If as a kid or as an adult you don't like the taste of Brussels sprouts, well, you probably learned that you could plug your nose because, well, they do taste kind of awful. But as you're chewing, molecules are going to break off the food and they're going to waft up into the nose. In this table, you're seeing the variation in sensitivity to a few different example molecules. The number on the right is the concentration in milligrams per liter that's needed before you can sense this substance. Ether is very difficult to smell. There needs to be quite a bit of it. Now, ether is interesting, as is chloroform, because they can knock you unconscious. So if you're working with either of these in the lab, and I've had to do this quite often, you want to make sure you're working in a fume hood, because by the time you smell them, you're probably getting pretty close to passing out. There's a few other interesting molecules on here that I'll mention. Things like oil of peppermint. These essential oils are quite often used to test a person's sense of smell. Butyric acid is found in vomit. It gives vomit its distinctive odor, and that's something we're quite sensitive to, which makes sense because if people around you are getting sick, then maybe you've ingested something bad as well, and that's something that your body would like to be aware of. Down the bottom there, methyl mercaptan is something we're very, very sensitive to. It is given off during rot. It smells like rotten cabbage. So of course we don't want to eat things that have that smell to them because they're probably going to make us sick. And also, interestingly, this is the molecule that they add to natural gas. Natural gas doesn't have an odor, but of course if there's a natural gas leak, that's very dangerous. So people very wisely long ago decided to start adding this compound to natural gas so that we can smell it and, take precautions not to light a match or to quickly turn off the source of the gas. But otherwise, without that, we wouldn't be able to smell natural gas. Incidentally, you might be wondering if ether and chloroform and unadulterated natural gas are so harmful, why can't we smell them? Well, they're not things that we would naturally come into contact with, so we never evolved that ability. Another interesting one is carbon monoxide. Carbon monoxide is also not something that's all that common, at least not in large concentrations in our natural environment. It's not something our ancestors would have had to worry about. And most above ground animals like ourselves are not sensitive to carbon monoxide. Even though it is very, very dangerous, we just don't sense it. Now, if you look at below ground animals, animals that burrow, take groundhogs and rabbits and things like that, they can detect carbon monoxide because that's something that's found underground and they are very sensitive to it. They get very, very worked up and agitated if they detect carbon monoxide. Okay, on to our last slide here to do with smell. I've mentioned this already, but olfaction is linked very intimately to memories and emotions. If you smell something, it can bring up a sudden emotional response, and also it will bring up memories of things that might have happened when you were last exposed to that smell, or if you were exposed to that smell even decades ago in childhood. And this makes sense because what your brain is doing is it's comparing that combinatorial code to the last time or previous times that it encountered that same code to try to figure out if it's something that's dangerous. So did it cause problems before, or is it something that might be a good thing? Just like olfaction, gustation, the sense of taste, relies on chemoreceptors. The tongue is a specialized organ. It does a few other things as well. It's involved in forming your food into a bolus that you can swallow easily. And it's also involved in making words. So it's involved in language. On the tongue, we have these little bumps known as papillae. At the base of a papilla, we have clusters of taste buds. And the taste buds have a pore that allows molecules to enter in and come into contact with gustatory epithelial cells. These will react to those molecules and trigger off an action potential that travels back to the brain. An average person has about 10,000 taste buds in total, and the taste buds wear out. They only last about 2 weeks. They're constantly being replaced and rebuilt. The ability to replace taste buds is something that suffers during aging, which is why elderly individuals may lose a lot of their sense of taste. But the taste buds are found at the base of papillae, and the taste buds contain these taste cells known as gustatory epithelial cells. Those cells have receptor sites, receptor proteins on the surface that will respond to different inputs. We can detect 5 different categories or modalities of taste. We can tell whether something is sweet or not. Sweetness is associated with something that has a high caloric value typically. Sugar is very easy to break down, and we evolved from primates that ate a lot of fruit. Fruit contains quite a lot of sugar, and this is one way we can detect when fruit is ripe and ready to be eaten. We can sense bitter. Bitter is associated with certain molecules. It depends on the shape of the molecule, And typically, things that are really bitter might potentially be harmful. A lot of toxic substances are bitter, so it makes sense that we would be able to detect these molecules. The sensation of sour is a result of high acidity or low pH. And that's something that's associated with unripe fruits. So again, from our ancient primate ancestors, we can tell using sweetness and sour when fruits are ripe. The taste that we associate with salty foods is due to the presence of sodium, of course, so we can sense how much sodium is present. And then finally, the fifth modality, which you might not be familiar with, is something known as umami. And this is a taste that's associated with high protein foods. It's a very kind of beefy sort of flavor, a very savory flavor. Now keep in mind that when you're eating, there's other things involved in that whole experience. Texture can play a big role. Temperature can change the sensation that you're experiencing. Smell is going to contribute to the subtleties of the flavors, and pain can be involved as well. If you're eating something spicy, that's actually triggering pain receptors within the tongue. We talked before about how capsaicin can trigger pain receptors. Capsaicin is found in a lot of chilies. It's a common myth that certain parts of the tongue are strongly associated with these certain modalities. That's not true. This came from some early studies that were debunked actually decades ago, but this myth seems to have stuck around. Now, there is a bit of variation in how these different receptors are distributed, and you can see that in the graph on the right, but you can see there isn't a strong difference between different parts of the tongue. All parts of the tongue can respond to these different modalities. The 5 different receptors that are responsible for the 5 different modalities respond to specific things. So the receptors that are responsible for sour respond to hydrogen protons. The receptors that are responsible for salt respond to sodium. The receptors that are responsible for sweet respond to sugars, and they do that based on the shape of the sugar molecule. The receptors that are responsible for bitter also respond to the shape of a variety of different compounds. And the receptors that are responsible for umami will respond to particular amino acids that are found in the food. It's been hypothesized, and there's pretty good evidence for this, that we might also be sensitive to lipid as a 6th modality. So it's possible that there's receptors that respond to the shape or some sort of input from lipids, and that's why we crave fatty foods. So once again, you can see that we have these taste pores that allow these molecules to move into a taste bud and come into contact with these gustatory cells. And note that a gustatory cell is going to synapse with a neuron that will carry an action potential to the brain. Sensory information from the taste buds travels through the vagus nerve and the glossopharyngeal nerve up to the thalamus. Remember that the thalamus is an important relay center for many of the sensations that we experience. From there, the information goes to the gustatory cortex, which is found in the insula and deep portions of the frontal lobe. Just like with smell, there's a great variation in how much of a certain compound it takes before we recognize it. What you're seeing in this table is the taste threshold for different compounds measured in micromoles per liter. It doesn't take much hydrochloric acid for us to recognize that as being sour. Hydrochloric acid, HCl, is a very strong acid. It gives off a lot of hydrogen protons. It takes considerably more sodium chloride or table salt for us to recognize that as a salty sensation. Now look at sugars, sucrose and glucose. It requires a lot of these molecules before they even register. Makes sense because this is a molecule that our body kind of craves, and we want a lot of it, so we don't experience that until we've got a lot of it. Saccharin is an artificial sugar, an artificial sweetener. And our receptors are far more sensitive to it. So it takes very little saccharin to register a sweet response. For bitter substances, it tends to take very little for us to reach that threshold. And that makes sense because, as I mentioned, a lot of the things that are toxic to us have a bitter taste. So it takes very little strychnine for you to taste that bad flavor in your mouth. Now I want to take a closer look at how these different sensory cells work. Remember that with olfaction, the sensory cells would detect certain shapes. Now that's what happens with bitter, sweet, and umami, but it's not what happens with salt and sour. If you eat something salty, you're going to have a lot of sodium dissolved within your saliva. And the cells that detect salt have channels that allow sodium to move freely into the cell. So if there's lots of sodium outside in the saliva, the sodium will move down these channels and depolarize the cell, causing it to release neurotransmitters. So that's pretty straightforward. Now with sour, hydrogen protons are going to be in high concentration in the saliva, and those hydrogen protons will bind to potassium channels. These potassium channels are normally open and they allow potassium to leave the cell. When a hydrogen proton binds to one of these channels, it causes the channel to close. Potassium is going to build up inside the cell, making it more positive on the inside, and that's going to cause a depolarization. With bitter, sweet, and umami, the receptors will sense the shape of the molecule, and that will activate a series of biochemical pathways within the cell. And the first step involves a G protein, as we've talked about with other receptors. This biochemical pathway will cause potassium channels to close, and that's going to increase the rate at which the cell will depolarize. But this pathway also involves activating calcium channels that will release calcium from the smooth endoplasmic reticulum. Also, there's going to be voltage-gated calcium channels that are going to be activated, and that's going to allow even more calcium to come in. And the end result, of course, is that with calcium present, the vesicles that are filled with neurotransmitter can bind to the presynaptic membrane and release their neurotransmitter. This diagram looks quite a bit different than the ones that you have in your notes. I decided to swap those out with this because this one's a bit more accurate, it's more up to date, it's more current with current research, and also it's a bit simpler. Now, one thing to note is that this diagram shows all the different receptors on one cell. That's just for convenience. That's not the way it works. We would have different cells that would just sense one thing. So we'd have cells that only did umami, cells that only did salt, etc. But this diagram's a really nice recap of what we've just talked about. So let's start with salt. On the cells that detect salt, we have these leak channels that allow salt to move down its concentration gradient. So they will move from the saliva, if you have a lot of salt in your mouth, into the cell. This is a case of facilitated diffusion. It's going to cause the cell to depolarize. When the cell depolarizes, voltage-gated calcium channels are going to open up and allow calcium in. Remember that calcium binds to cadherins. These are proteins that are found on the surface of vesicles. That allows the vesicles that contain neurotransmitter to bind to the presynaptic membrane and dump their contents into the synaptic cleft. The neurotransmitters will move across the cleft, bind to receptors on the neuron, cause it to depolarize, and send off an action potential back to the brain. And for all of these receptors, those last few steps, from the voltage-gated calcium channels to the release of neurotransmitter, they're all the same. We have the same steps that bring about the release of neurotransmitter. Okay, for sour, we have hydrogen protons that will bind to potassium channels, cause them to close. Potassium can't leave. That's going to cause the cell to depolarize. And again, calcium will rush in. That'll cause the release of neurotransmitter. With bitter, sweet, and umami, Each of those cell types has a receptor that will only bind molecules of a certain shape. If that happens, that's going to set off multiple signal transduction pathways, and these pathways are going to be initiated by G proteins. These pathways are going to cause potassium channels to close, and that will help bring about a depolarization. At the same time, though, calcium is being released from internal stores, calcium that's stored within the smooth ER. That, coupled with calcium that's going to rush in as the cell depolarizes, will cause the release of neurotransmitter. Now, one thing that's kind of interesting I haven't mentioned is that we've only known of the existence of umami receptors in humans since 2002. It's not that long ago. One of the ways you can trick umami receptors is with artificial glutamate. Glutamate is a form of glutamic acid, and glutamic acid is the amino acid that the receptors normally respond to. Monosodium glutamate binds to umami receptors, and it causes food that has it added to taste more savory. So even though we haven't known about the umami receptors for very long, we've been adding MSG to food for decades to make it taste a bit beefier. There are many other fun ways to trick your taste buds. In Africa, there's a plant that produces berries known as miracle berries or miraculum berries. These berries contain a substance known as miraculum. Miraculum will bind to your sweet receptors. It has a similar shape to the sugar molecules that would normally bind. But there's a few things that are unusual about this. First of all, sugar doesn't stay bound for very long, just for milliseconds. So it will cause an action potential and leave the receptor. Miraculum, on the other hand, can stay bound to a receptor for over an hour. It doesn't set off the receptor though. It just binds to it and blocks that cleft that sugar would normally bind to. So if you take one of these berries or an extract of the berry, chew it up and swish it around in your mouth and make sure that it coats your tongue, and then take a teaspoon of sugar, it won't taste like anything. It won't taste sweet. And that's because this miraculum is blocking the sweet receptor. Now this is where it gets interesting. If you take something very acidic, like some vinegar, or you suck on a lemon, that is going to cause the denaturation of that miraculum. It'll stay attached to the sweet receptor, but it will change shape and it will trigger the sweet receptor. And now all you can taste is sweet. So sucking on something that is acidic will cause a sweet sensation. And again, the reason it does that is because the miraculum changes shape. Now you can play around with this yourself. It's totally safe. You can buy it on Amazon. It's not cheap, but give it a try. There are numerous other artificial sweeteners that mimic the shape of sugar. Unlike miraculum, they behave just like sugar as well. They bind for a fraction of a second and then release. Many of these artificial sweeteners are far more effective at binding than is actual sugar, so you don't need to use nearly as much of them to get the same effect. And now we're going to look at our last special sensory organ for the day, and this would be the ear. The ear is responsible for two things, two separate senses. We have hearing, of course, and that information is going to be processed in the auditory cortex of the temporal lobe, but we also have equilibrium, which is processed in the cerebellum. There are different organs within the ear that process these two sensations, and the receptors in both cases are mechanoreceptors. Broadly speaking, we can divide the ear into three regions. We have the outer or external ear, that's the floppy bit that hangs off the side of your head, and the canal that leads to the eardrum or tympanic membrane. And then we have the middle ear, which contains the ossicles the little tiny bones that vibrate within the ear. And then we have the inner or internal ear, which contains the sensory receptors. The part of the ear that sticks out beyond your head is known as the auricle or pinna. Its function is to collect sound and channel that sound into the external auditory canal. It's supported by elastic cartilage. The external auditory canal is a chamber that passes from the outside world through a hole in the temporal bone, and it ends at the tympanic membrane. Now that hole in the bone is referred to as the auditory meatus if you're British, or the auditory meatus if you're American. But if you look at some textbooks, they'll refer to that entire fleshy canal as the meatus. The canal is lined with skin, and the skin is going to contain ceruminous glands, which are wax glands. The wax is there to help protect the skin, keep it moist, protect the ear from dust and debris, and also the wax will help prevent the growth of bacteria. The middle ear is an air-filled cavity within the temporal bone. It only plays a role in hearing, not in equilibrium. It's going to conduct vibrations from the tympanic membrane to the inner ear. There's an auditory tube that connects the middle ear to the throat. It's also known as the eustachian tube, and this is there to allow equalization of pressure outside and inside the middle ear. So it will equalize the pressure within the middle ear with the external environment. Now, as you know, if you're flying on a plane, the cabin pressure is going to drop slightly, and at some point you'll feel your ears pop, and that's the pressure being equalized. You can speed this up by yawning or swallowing, because that's going to stretch the tube, and the tube is usually collapsed. If you have a bad cold and this becomes filled with mucus, it can be much more difficult to equalize the pressure. Incidentally, in children, just because of the way their skull grows, the eustachian tube tends to have a bit of a bend in it, and it tends to collect fluid in this bend, and they can have more trouble getting that pressure equalized. And that results in them having more trouble on plane flights. The middle ear contains the ossicles. Ossicle in Latin means tiny bone, and that's exactly what these things are. They're the three smallest bones in your body. Right up against the tympanic membrane is a little tiny bone known as the malleus. Malleus is Latin for hammer. That connects to a bone known as the incus. Incus is Latin for anvil. And then finally we have the stapes, which is Latin for stirrup. All of these bones look a little bit like what their name suggests, especially the stapes. What these bones do is they transfer movements of the tympanic membrane to the inner ear. These bones are clearly very tiny, but my statement that they're the smallest bones in the body isn't 100% accurate. They're definitely the smallest bones that have definitive shapes and definitive names, but you do have some smaller sesamoid bones within your hands and feet. Now let's look at the inner ear. This is where all the magic happens. This is where the sensory cells are located. We have two parts to the inner ear. We have the cochlea, which is a snail-shaped structure. It's responsible for hearing. And then we have the semicircular canals, which are responsible for equilibrium. All of these structures are filled with fluid, and they're surrounded by a casing of bone. And that casing of bone is referred to as the osseous labyrinth. Osseous meaning bony, labyrinth meaning maze. The diagram on the upper left shows you the cochlea straightened out, unraveled. So imagine if we took that tube and we bent it back on itself into a spiral, like a snail shell, then we would have the cochlea as it actually appears in life. Now the reason they've done this, the reason they've unraveled it, is it makes it a lot easier to understand what's going on. We have sound waves that travel through the air. They're pressure waves, and they hit the tympanic membrane, and they push on the tympanic membrane. The result is that the tympanic membrane moves back and forth. It's going to transfer that movement to the malleus, and then onto the incus, and then onto the stapes. The stapes, as you can see in this diagram, is pressed up against a membrane known as the oval window. On the other side of that window, we have a fluid known as perilymph, and that fills most of the cochlea, as we'll talk about in a bit. The movement of the stapes pushing against that oval window will create pressure waves through this perilymph. Basically, the stapes acts like a little tiny piston. The pressure waves move around the top chamber and then back along that bottom chamber. Those chambers are continuous with each other. Those pressure waves eventually get to the round window, and this is a membranous window that will allow for the release of excess pressure within the system. Note that as the waves travel around the cochlea, they distort membranes in the middle. And those distorted membranes are going to rub up against sensory cells, as we'll see. The diagram down the bottom is showing you the function of the middle ear. So we have this sound energy that comes in, and it's collected by the tympanic membrane. Tympanic membrane is a fairly large membrane compared to the oval window. So we're collecting energy over a large surface, and then we're transferring that energy to a smaller surface. We're transferring it to the oval window. And by doing that, we amplify the sound. We amplify the energy in that signal. So the function of the middle ear is amplification. So again, we have vibrations of the stapes that are going to push and pull on that membranous oval window, and that's going to cause pressure waves within the perilymph. Again, remember that we collect energy over a large surface area, and we focus that onto this small surface area. The pressure waves are going to travel through this upper chamber known as the scala vestibuli. They round the corner and they come back along the lower chamber known as the scala tympani. The diagram on the left is showing a transverse section through that straightened out cochlea. We have our chamber at the top, the scala vestibuli, and then our chamber at the bottom, the scala tympani. The pressure waves would be moving towards us along this scala vestibuli and then heading back away from us through that scala tympani. Now there's a third chamber that runs in between these two chambers, and that's known as the cochlear duct. Those other two chambers contain perilymph, but the cochlear duct contains endolymph. They're pretty similar. It's more to do with location. Within that cochlear duct, we have an organ known as the organ of Corti. And this organ runs the length of that cochlear duct. And you're seeing that up close to the right. Note that we have this basilar membrane, and then attached to that we have some support cells, and embedded within those support cells we have the actual receptor cells. They're referred to as hair cells. And that's because they have these projections that stick out the top that superficially resemble hairs. They're not actually hairs, but we'll talk about that in a moment. Above that, we have a membrane known as the tectorial membrane. And then above that, we have the vestibular membrane that lines the scala vestibuli. Well, as pressure waves move through this system, they distort the membranes and they cause the hair cells to rub against the tectorial membrane. What's going to happen next is that these cells are going to be triggered. They're going to send off action potentials, and the action potentials will travel down the cochlear nerve to the cochlear vestibular nerve and then back to the brain. The diagram on the right shows sound waves entering through the ear canal, hitting the tympanic membrane. That movement is transferred to the stapes, and the stapes is going to create pressure waves within the perilymph. As those waves move along the interior of the cochlea, they're going to distort the vestibular membrane. Now, that cochlear duct in the middle contains fluid, so distortion of the vestibular membrane is going to move that fluid, and it's going to distort the underlying tectorial membrane. And that membrane is going to rub against those hairs, which, as I said, are not actually hairs. They're properly referred to as stereocilia, although they're not really quite cilia either, but we'll get there in a second. The diagram, or actually photograph, electron scanning micrograph, on the left shows you how these stereocilia are arranged on these hair cells. They're arranged in these little V-shaped or chevron patterns. The top of the hair cells contain these stereocilia. And as you can see on the diagram, they're not actually hairs, and they're also not really cilia. They're projections of the cell that contain cytosol that's continuous with the cytosol of the cell body. What's really fascinating is that there are mechanical gates at the top of these projections. So right at the tip, we have these channels that have a physical trap door on the top of them. The trap doors are tethered together. There's connective fibers that run from one trap door on one stereocilia to the next trap door on the next stereocilia. And if these hairs or projections rub up against the tectorial membrane, it causes all of these trap doors to open together. This allows potassium to rush in. So those trap doors sit atop of specialized potassium channels. Potassium rushes in, and that causes depolarization of the cell. That's going to activate voltage-gated calcium channels. Calcium rushes in. That allows vesicles to bind and dump neurotransmitter into the synapse. The cells that are responsible for the sensation of equilibrium are very similar to the cells that are found in the cochlea that are responsible for hearing, and they work in a similar fashion. Let's take a look at equilibrium. There's two types of equilibrium. We have static equilibrium and dynamic equilibrium. Static means non-changing or non-moving, and this refers to the sensation of how your head is positioned relative to the downward force of gravity. Are you upright? Are you upside down? Is your head tilted to the side? Dynamic means changing or moving. This refers to the sensation of acceleration. Now, you can't sense constant velocity, but you can sense a change in velocity. If you're traveling along in a car and you slow down, or you speed up, or you turn, change directions, you can sense that, and this happens within the base, the ampulla of the semicircular canals. So that feeling of static equilibrium, which way is up, which way is down, that's being produced within this large structure here known as the vestibule. And then the sensation of dynamic equilibrium, accelerations, are being generated within these structures here, which are known as ampulla. Let's start by taking a look at how static equilibrium works. Within the vestibule, we have areas known as maculae that contain the hair cells that act as our sensory cells. Again, they have a structure that's very similar to the hair cells that are found within the cochlea. They're going to provide information on the position of the head and send that information via the vestibular nerve, which of course will fuse with the cochlear nerve to form the vestibulocochlear nerve. We have our hair cells, and they're surrounded by support cells, and the hair tufts are embedded into what's called the otolithic membrane. This is a gel substance, And on top of that gel, we have otoliths, which are tiny stones. Well, technically speaking, they're crystals of calcium carbonate. And they add a little bit more weight to that gel layer. And this is perhaps a better diagram of that. Again, we can see our supporting cells, we can see our hair cells that are attached to vestibular nerve axons. We can see this jelly layer, which makes up the otolithic membrane, and then we have our otoliths sitting on top. When you tilt your head, that's going to cause this gel to flow. And the gel and the otoliths are going to cause the hair cells to bend. And just like happened in the cochlea, when the hair cells are bent, they open up little trap doors at their tips, and that causes potassium to rush in and depolarize the cell. This maybe isn't the clearest image of this, but this is what this layer looks like under a light microscope. You can see that we have otoliths at the surface, we have this thick gelatinous layer, we have hairs of hair cells embedded into that layer, and they're a bit difficult to see. And then at the bottom there we have our actual epithelium that contains the bodies of the hair cells and also the supporting cells. Dynamic equilibrium is a result of the motion of fluid within the semicircular canals relative to the body. And I say that because generally it's the body that's moving and the fluid is staying still. But of course, the way physics works, it doesn't matter which thing is moving. But we'll come back to that. The semicircular canals are arranged in the three planes of section: frontal, transverse, and sagittal. Or perhaps a better way to think of it, they're at right angles to each other and they're in the X, Y, and Z planes. At the base of each of the semicircular canals, we have a swelling known as the ampulla. And within the ampulla, we have collections of hair cells that make up a structure known as a crista ampullaris. It's basically just a little mound or ridge of these sensory cells. The hairs of the hair cells are embedded within this rather tough gelatinous cap that's known as the cupula. Now, if we have movement of the ampulla relative to the fluid, or vice versa, that can cause this cupula to be moved, to bend over and bend the hair cells. In this example here, the body is accelerating towards the left. The body is going to accelerate first. It's going to take a little bit of time, a tiny bit of time, for the fluid, the endolymph, to catch up. And that means the endolymph is going to push on the cupula. So you can think of this as the fluid standing still and the body moving, which is actually what's happening. Or if you want to think of it as the fluid moving across the cupula, so it's moving relative to the cupula, doesn't matter, same effect. It's going to bend the cupula over, that's going to bend the hairs, and that's going to cause these cells to fire off an action potential. They're going to be depolarized in the same way that we've already talked about. The little trap doors on the top of those mechanoreceptors are going to open, and that's going to allow potassium to flow into the cell and to depolarize it. Here you can see one of those cristae, and there's hair cells embedded within it, and on top of this we have a little hat or cap, which is the cupula. And the big space at the top, that would be the interior of the ampulla, and it's filled with endolymph. Here we're seeing the structures of both the vestibule and the ampulla in the same image. So at the top there we have a semicircular canal coming down. We have a crista sitting within our ampulla. And then under that, now we're looking into the vestibule, and we can see our otoliths on top of the macula. And again, the macula is a collection of hair cells. To summarize, static equilibrium is the result of gravity pulling on this viscous gel known as the otolithic membrane. And I just realized I've been saying otolithic, which is kind of silly. I'm just kind of tired. It's the otolithic membrane, and it has otoliths on top of it to add a little more weight. When you tilt your head, that viscous fluid moves and it bends over the hair tufts and triggers the cells. You can keep your head tilted for a long time, and because that material is so viscous, you'll still continue to have that sensation. In dynamic equilibrium, we're talking about accelerations. So basically, sudden movements of the body will cause endolymph to slosh around within the semicircular canals. When the fluid moves down into the cupula, or if the cupula is moving relative to the fluid, then that's going to bend the structure over and bend the hair cells, and that will trigger off action potentials. Now, this could be due to your body accelerating or decelerating or changing the direction of its movement, or it could be due to rapid motions of the head. So if you move your head, of course, you have to accelerate it to initiate that motion. And that's the end of our topic and the end of the course. So thank you very much for listening. I hope you enjoyed the course, and you've got a few weeks now where you don't have to hear my voice. So enjoy your holidays, and maybe I'll see, or at least talk to some of you in 112 next term.